Hi there. Welcome to Labster. Here you will learn how to design an experiment by defining a scientific question, making a hypothesis, and planning the experiment. Will you be able to design an experiment that can identify the cause of a recent epidemic? This simulation uses voiceovers. If you don't have audio, click the speaker symbol on the top right of your lab pad to switch the play mode. If you disable the sound effects, the lab pad will slide up whenever there are instructions to read. You can select what type of audio you prefer. Deselecting any of the options will mute that option. Click Continue to learn how to navigate through your mission. Using the lab pad is an important way to get the most out of this simulation. Is this the first time you've played a Labster simulation? Perfect, then let's get started with your first mission. I am Dr. One, your AI lab assistant. When I am not teaching students, I have developed a habit of scanning our medical databases for anomalies. I think I might have made an interesting finding, but I need a human's help now. Let me share some information with you. There has been a spike in patients reporting with headaches, anemia, and high blood pressure. My investigation has shown that these symptoms are caused by kidney injuries. I have compared reports from several national institutions and found something very interesting. There are more reports in some countries than in others. The percentage of reported cases isn't evenly distributed. What's interesting is that this distribution correlates suspiciously well with the sales revenue of a recently introduced medication, XP. XP is heavily advertised as a tonic and restorative medication in all of the affected countries. It turns out that XP contains 3 active ingredients. None of the 3 compounds is known to cause health issues. Let's go to the lab to find out if this correlation is just a coincidence. I need your help to figure out if my suspicion is right. A lot of people might be unknowingly poisoning themselves. We need to investigate this. Before we get started, pick up a lab coat to put it on. Almost ready. Now, find a glove box and click on it. I have prepared some microscopy slides with cross-sections of different kidney biopsies. Would you like to take a look at the samples under the microscope? Awesome. I can show you the slides at the microscopy workbench. Click on it to go there. Pick up the slide containing healthy human kidney tissue and place it on the stage of the microscope by clicking on the microscope, then the microscope stage. Click and hold the black arrows on the microscope screen to find the large round structure. Whenever any of the arrows on the side of the screen shows up in black, click on it to move to that side. Would you like to try to find the structure by yourself? Click on the lab pad for more information. Observe the tissue structure of a healthy kidney tissue. Can you identify the large round structures? They are glomeruli, capillary loops through which plasma is filtered before being processed in the tubules into urine. The tubules are the predominant structure. Tubules consist of a simple layer of epithelial cells that form a tube. There is a third distinct structure. Have a look at the slide and find the black arrows that indicate it. What are the arrows pointing at? Raise the lab pad to find out. Roses are red. And so are blood vessels. You can find a blood vessel containing red blood cells. Let's have a closer look at it. You can zoom into the slide by switching to an objective with a higher magnification. Try it. Click on the 100 times objective to take a closer look at the slide. Well done. At this magnification you can clearly see the nuclei of the individual cells. Feel free to keep exploring the healthy kidney tissue under the microscope. When you are done, remove the healthy sample slide and replace it with the kidney biopsy of the patient taking XP as medication. Oh, look at this. The tubules are grossly and irregularly dilated, and the lining epithelium is flattened. This is a sign of tubular injury. After observing the slide, switch to the higher magnification to see more detail. At this magnification you can see that the tubules are shrunken and the epithelial structure is unrecognizable. The tubular lumina are filled with dense, eosinophilic material, representing sloughed epithelial cells and protein. These collections of cells and protein can form solid casts of the lumen, which are then excreted and can be identified by analyzing a urine sample under the microscope. Have a look at the slide and find the black arrows indicating another symptom. The arrows indicate dense, shrunken nuclei. These symptoms suggest that XP patients are suffering from a disease called analgesic nephropathy. Check out the theory if you want to learn more about this disease. Well, we seem to be able to make the observation that is a correlation between XP sales and analgesic nephropathy. We need to conduct some more analyses before making any conclusions. Go to the next room to continue. You have already made an observation, which is the first step in the scientific process. Click on the next step in the scientific process. You can observe the entire scientific method by clicking on the View Image button. What is the most pressing scientific question to address, in order to tackle this public health problem? That's it. If XP is the cause of this epidemic, we need to stop it as quickly as possible. The only way to do that is by generating sound scientific results. Now that we have defined our question, we need to state a hypothesis. What is a hypothesis? Well done. A hypothesis is a tentative, testable, and falsifiable explanation for an observed phenomenon. Make sure you don't confuse the predictions with the hypothesis. Predictions are derived from the hypothesis, but the two are not synonymous. The data from the National Health Databases suggests that XP might be the cause of the observed kidney injuries. We need to come up with an experiment to test this hypothesis. We need to define the experimental variables. Let's go to the model workbench and figure out how to plan the experiment. Go to the model workbench. Hover over the different objects and identify the best experimental model to answer our question. Before we can conduct an experiment in mice, we need to get an approval from the Ethics Commission. I will write the proposal and send it to them. Let's speed up the time while we are waiting. Let's wait for approval from the Ethics Commission. Finally, the Animal Experimentation Ethics Committee's response came back to us. They did not approve the proposal. Their reply states, "You were unable to justify the use of animals when you could have used cell lines instead." Bummer. We wasted 2 months waiting for their reply just to get a rejection. We should have gone for the cell lines instead. Have a look at the available human cell lines in the incubator. There are 4 cell lines readily available in our lab. Which cell line would you use for the experiment? Good thinking. The patients suffer from kidney injuries, hence, we should use kidney cells to test the compounds. I have extracted the 3 compounds from the drug XP and prepared them on the experimentation workbench. Pick up the kidney epithelial cell culture in the incubator and bring it to the experimentation workbench. Place the cell culture plates on the workbench. One of the variables for this experiment is the concentration of each of the compounds. We would need to prepare a plasma solution with a concentration that would be found in the bloodstream. To do this, we would need to prepare samples by mixing each of the 3 compounds with buffer using a pipette. This way, we can prepare samples to test on the kidney endothelial cell cultures and observe the effects. Remember, we are only planning the experiment. The process may have to be adjusted if this does not go to plan. Pick up a kidney epithelial cell culture plate and place it on the workbench. If XP is responsible for the recent epidemic, we would expect that the kidney epithelial cells die as a result of XP exposure. Let's define the variables of our experiment. The number of dead cells is one of the variables in our experiment. What type of variable is it? Awesome. The number of dead cells is our dependent variable. It's the measurable outcome of the experiment. What would be the independent variable? Marvelous. We can expose the kidney cells with different concentrations of the compounds, the independent variable, and observe if the cells are dying, the dependent variable. Now that we have defined our experimental variables, we can state our research hypothesis. If XP causes kidney injuries, and I expose kidney epithelial cell cultures with XP, then the kidney epithelial cells will die off. Great, we have completed the final step before we are ready to conduct our experiment. Let's share our hypothesis and experiment plan with Professor Miller.
Thanks a lot for joining us so spontaneously. There is a growing interest in the XP case. I would love to hear what you have found out so far. I heard you already came up with your hypothesis for this research experiment? Interesting. So your results are not conclusive yet. What needs to be done to prove that XP is responsible for the epidemic? That makes sense. A single experimental sample is not proof. We can only make conclusions if the experiment is reproducible and the results are consistent over several replicates. Once you have performed the replicates, what are you going to do? Great. I can't wait to hear back from you. Thank you so much for your thorough investigation. Your research will improve the health of millions of people. Keep up the great work.
Great work. We were able to follow the scientific method to plan this experimental investigation of the XP drug. We conducted our background research, chose an experimental model, defined our variables, and came up with a hypothesis. Professor Miller is very excited to move on to experimental research and gain conclusive results. Let me ask you one last question. Do you feel like you gained relevant knowledge during this simulation? Thank you for your feedback. Your score has been saved together with your 100% progress. Congratulations, you have completed the simulation. We hope you enjoyed it and you feel wiser now.